Chemotherapeutic agents are antimicrobial drugs that fight disease by targeting a particular structural difference between human cells and those of the microbe. Because bacteria have many such differences, it's much easier to develop antibacterial drugs. Viruses depend on the host cell's machinery, so there are fewer differences to target. And eukaryotic pathogens, such as fungi and protozoa, have fewer differences from eukaryotic human cells, making it difficult to find drugs that have selective toxicity against these pathogens. Antibacterial drugs have several different modes of action. One of the ways in which antibacterial drugs can kill bacterial cells is to block the formation of bacterial cell walls. Normally, the peptidoglycan layers are held together with peptide crosslinks. Penicillin and its relatives block the formation of these peptide crosslinks. This weakens the cell wall, ultimately causing the cell to lyse. Antibacterial drugs such as polymyxin attach to phospholipids in the bacterial cytoplasmic membrane and interfere with its integrity. Eventually, the cell lyses. Because this mode of action also interferes with human cell membranes, this antibiotic is used only on the skin, where the outer layers of cells are already dead. Antibacterial drugs such as tetracycline, chloramphenicol, and streptomycin kill bacteria by inhibiting protein synthesis. Tetracyclines block the docking site of the transfer RNA, preventing amino acids from being added. Chloramphenicol prevents formation of peptide bonds between amino acids. Macrolides like erythromycin block the movement of messenger RNA through the ribosome. Aminoglycosides like streptomycin cause a change in the shape of the smaller 30S ribosome subunit, resulting in misreading of messenger RNA and insertion of incorrect amino acids. Bacteria have certain metabolic pathways that are not found in humans. One of these is the pathway for synthesis of folic acid, a coenzyme used in the synthesis of nucleic acids. Humans do not have such a pathway and require folic acid as a dietary supplement. One of the substrates in this pathway is para-aminobenzoic acid, or PABA. Antibiotics such as sulfonamides or sulfa drugs block a step in the folic acid synthesis pathway by acting as a competitive inhibitor to PABA. This prevents synthesis of folic acid and therefore synthesis of nucleic acids. The bacterium is unable to reproduce. Most organisms synthesize nucleic acids in a very similar manner, so it's much more difficult to find differences to target for these pathways. However, there is a group of synthetic drugs called quinolones and fluoroquinolones that block DNA gyrase in bacterial DNA. They have little effect on eukaryotic cells. Other drugs, such as rifampin, bind more readily to RNA polymerase in prokaryotes than eukaryotes and can be used to block transcription.